Hello, hello. Hello and welcome back to आजको लाइभमा. ओके, ठिक छ, यता पनि चलिरहेछ ल. सो हामी चाहिँ आज वाच पार्टी गराउँदैछौँ लाइभ नोबेल प्राइज इन फिजियोलोजी अर मेडिसिन 2024 को. सो give me a minute. So anyway, आजको चाहिँ हामी लाइभ स्ट्रिम वाच पार्टी गर्दैछौँ एन्ड वी विल वाच the Nobel Prize lecture in Physiology or Medicine 2024 together. अनि त्यसपछि हामी लाइभ कमेन्ट्री त के भन्नु हामी हेर्छौँ लाइभमा एन्ड देन we'll just do that. So that's it. अनि let me do something. अनि सो हामी कति मिनटमा हो? 3 minutes. Okay, in 3 minutes Nobel Prize को lecture physiology and medicine सुरु हुन्छ. Before that हामी चाहिँ के गर्छौँ भन्दाखेरि नोबेल प्राइज फिजियोलोजी र मेडिसिनमा किन दिएको चाहिँ त्यो हेर्छौँ है. नोबेल प्राइज 2024 मेडिसिन हामी त किन दिएको थियो त्यो एउटा ब्रिफमा हेर्छौँ अनि देन हामी वाच पार्टी सुरु गर्छौँ. राइट? अलराइट, ठिक छ. ओके, properties. Okay, so this is Nobel Prize in Physiology or Medicine 2024. दुईटा winners हाम्रो Victor Ambros and Gary Ruvkun. And the Nobel Prize in Physiology or Medicine 2024 was awarded jointly to Victor Ambros and Gary Ruvkun for the discovery of microRNA, mRNA and its role in post-transcriptional gene regulation. यो भनेको के भन्दाखेरि दुईटा चिज हुन्छ, डिएनए र आरएनए. डिएनए भनेको डिओक्सिराइबोन्युक्लिक एसिड, आरएनए भनेको राइबोन्युक्लिक एसिड. अनि के हुन्छ भने डिएनएसँग इन्फर्मेसन हुन्छ कुन चाहिँ यो एसिटिजी चार्ट प्रोटिन हुन्छ क्या हाम्रो एसिटिजी यो चार्ट प्रोटिन मिलेर डिएनए बनेको हुन्छ. डिएनएमा यो इन्फर्मेसन हुन्छ कि कुन प्रोटिन पछि कुन चाहिँ प्रोटिन बाइन्ड हुन्छ, सिक्वेन्समा कुन चाहिँ आउँछ, सो द्याट नयाँ बन्ने सेल अथवा नयाँ बन्ने प्रोटिन बडीको जति पनि बायोकेमिकल प्रोसेस हुन्छ त्यो सबै बायोकेमिकल प्रोसेसमा कुन चाहिँ बायोकेमिकल बन्नको लागि यो अर्डरमा डिएनएले यसरी पनि यसरी बनाउनु पर्छ भनेर डिएनएसँग इन्फर्मेसन हुन्छ. अनि आरएनएले के गर्छ भन्दाखेरि त्यही डिएनएको इन्फर्मेसनलाई ट्रान्सक्रिप्ट एज अ ट्रान्सक्रिप्ट लिएर चाहिँ उसले त्यो उसले के गर्छ भन्दा डिएनएलाई लिन्छ, उसले ब्रेकडाउन गर्छ अनि त्यसपछि प्रोटिनलाई अनि पढ्दै अनि अट्याच गर्दै जान्छ अनि कपी बनाउँछ, अर्को कपी बनाउँछ अनि त्यसपछि फेरि रिअट्याच गरिदिन्छ अनि नयाँ ठाउँमा पठाइदिन्छ. नयाँ ठाउँमा पठाएपछि अब नयाँ सेल बन्ने हो, वाट एभर त्यो बायोकेमिकल के बन्ने हो त्यो हुन्छ. सो mRNA को रोल इन इट्स पोस्ट ट्रान्सक्रिप्सनल जिन रेगुलेसन त्यसैको लागि दुईजना हाम्रो नोबेल प्राइज विनरहरूलाई अवार्ड दिएको थियो। सो आज चाहिँ उहाँहरूको अवार्ड सेरेमनी सायद हुन्छ होला, मलाई ठ्याक्क थाहा भएन, बट लेक्चर चाहिँ हामीले आज पाउँछौँ। सो वी विल वाच इट टुगेदर एन्ड आई थिङ्क अब चाहिँ 1 मिनट जति छ होला, या 23 सेकेन्ड। सो लेट्स बिगिन है ल। मैले यहाँ गरिदिन्छु ल, म आफूलाई चाहिँ यहाँ राख्छु so that we can have the action together. ठिक छ। ए ओके, त्यसो भए मलाई सानो बनाउनु पर्यो नि त। यहाँ राखौँ। ओके, सो लाइभ इन 7 minutes। ए ओके, एक्सटेन्ड भइसकेको छ, फेरि एक्सटेन्ड भएको र आई डोन्ट थिङ्क सो। 18:30 यस, अब सुरु हुनुपर्ने हो। इट सुड बिगिन एनी मिनट नाउ। एनिवे वी विल वेट। सो हामी के कुरा गर्दै थियौँ भन्दाखेरि नोबेल प्राइजकै लेक्चरको बारेमा कुरा गर्दै थियौँ एन्ड press release अथवा advance information, yes, press release Samsung. This year's Nobel Prize honors to science for the discovery of a fundamental principle governing how gene activity is regulated. The information stored within our chromosomes can be likened to an instruction manual for all cells in our body. As हामीले भन्यो नि, डिएनएमा हाम्रो इन्फर्मेसन, हाम्रो मेसेज हुन्छ, त्यो मेसेज, अनि त्यो मेसेजलाई चाहिँ कसरी ठ्याक्क। So here is the thing. Every cell every cell contains same chromosome. So every cell contains exactly same set of genes and exactly same set of instructions. Yet different cell types such as muscle and nerve cells have very distinct characteristics. How do these differences arise? The answer lies in gene regulation, which allows each cell to select only relevant instruction. This ensures that only correct set of genes is active in each cell type. जस्तै मेरो यो यहाँ मेरो सपोज यहाँ मसल छ भने यहाँ मसलमा र यहाँ अर्को नर्भ सेल छ भने यो नर्भमा जिनको इन्स्ट्रक्सन एउटै हुन्छ, खालि फरक के भन्दाखेरि कुन चाहिँ जिन एक्टिभ भयो भने पर्टिकुलर टाइपको सेल बन्ने, अर्को टाइपको जिन एक्टिभ भयो भने अर्को टाइपको सेल बन्ने, त्यो चाहिँ केले गराउँछ भन्दाखेरि mRNA ले गराउँछ। सो अब नेक्स्ट प्याराग्राफमा अर्को इन्फर्मेसन छ। Victor Ambros and Gary Ruvkun were interested in how different cell types develop. They discovered microRNA, a new class of tiny RNA molecules that play a crucial role in gene regulation. Their groundbreaking discovery revealed a complete new principle of gene regulation that turned out to be essential for multicellular organisms, including humans. It is now known that the human genome codes for over 1000 microRNAs. Their surprising discovery revealed an entirely new dimension to gene regulation. Dimension, very Sadhguru yaad dimension get if you watch Science is Dope. So anyway, not extrapolate I mean discussion microRNAs are proving to be fundamentally important for how organisms develop and function. So as we said, yeah, information picture the flow of genetic information from DNA to mRNA to proteins. Identical genetic information is stored in DNA of all cells in our bodies. This requires precise regulation of gene activity so that only correct set of genes is active in each specific cell type. Human genome contains nearly 20,000 genes. जिनमा डिएनए हुन्छ, डिएनए ट्रान्सक्रिप्सन एमआरएनए गर्छ, एमआरएनए ट्रान्सक्रिप्सन भइसकेपछि एमआरएनएमा जान्छ, एमआरएनएले ट्रान्सलेसन गर्छ, ट्रान्सलेसनबाट प्रोटिन बन्छ, त्यो प्रोटिन स्पेसिफिक सेल टाइप बनाइदिन्छ। सो मसल फाइबर हो भने मसल बनाउने मात्र जिन्स एक्टिभेट हुन्छ। नर्भ फाइबर हो भने नर्भ बनाउने जिन्स मात्र एक्टिभेट हुन्छ। हुन त सबै सेलमा सेम सेट अफ जिनको इन्फर्मेसन हुन्छ, बट कुन चाहिँ एक्टिभ गराउने भन्ने कुरा mRNA डिसाइड गर्छ। ओके, अनि जेनेटिक इन्फर्मेसन फ्लोज फ्रम DNA to messenger RNA via a process called transcription, and then onto the cellular machinery for protein production. Their mRNAs are translated so that proteins are made according to the genetic instruction stored in DNA. Since the mid-20th century, several of the most fundamental scientific discoveries have explained how these processes work. Our organs and tissues consist of many different cell types, all with identical genetic information stored in their DNA. However, these different cells express unique set of proteins. How is this possible? Answer lies in the precise regulation of gene activity so that only one correct set of gene is active in a specific cell type. Okay. सो एउटा इक्जाम्पल छ, जस्तै मसल सेल अथवा इन्टेस्टाइनल सेल एन्ड डिफ्रेन्ट टाइप्स अफ नर्भ सेल, उनीहरूले चाहिँ आफ्नो उनीहरू स्पेसिफिक टाइपको सेल हुन् जसले चाहिँ स्पेसिफिक टाइपको फङ्सनिङ गर्छ होइन। त्यो गर्नको लागि जिनको एक्टिभिटी पनि मोस्टली फाइन ट्युनिङ गराउनु पर्छ सो द्याट त्यो सेलुलर फङ्सनको लागि एडाप्ट हुन सकोस् होइन। अनि हाम्रो बडीमा डिफ्रेन्ट फिजियोलोजिकल कन्डिसन्सले गर्दा हाम्रो बडी पनि चेन्जिङ हुन्छ डिफ्रेन्ट कन्डिसन्समा। सो यस्तो गर्दाखेरि यदि जिन रेगुलेसनलाई मेन्टेन गर्न सकेन भने सिरियस डिजिजेसहरू लाउन सक्छ, जस्तै क्यान्सर, डायबिटिज अर अटो इम्युनिटी। अटो इम्युनिटी भनेको के भन्दाखेरि हाम्रो बडीले आफ्नै बडीलाई नचिनेर डब्लुबिसीले आफ्नै बडीका सेल्सहरूलाई पनि अट्याक गरेर डिस्ट्रोय गर्न थाल्छ। सो देयरफोर अन्डरस्ट्यान्डिङ द रेगुलेसन अफ जिन एक्टिभिटी हेज बिन इम्पोर्टेन्ट रोल फर मेनी डेकेड्स। सो ती डेकेड्सदेखि नै यो प्रब्लमलाई चाहिँ वी वर ट्राइङ टु सल्भ, अनि दिस टु साइन्टिस्ट फाइनली डिस्कभर्ड त्यो mRNA को प्रोसेस, एन्ड देन बिकज अफ द्याट उनीहरूलाई चाहिँ यो नोबेल प्राइजको 2024 को अवार्ड दिएको थियो। सो हामीले के बुझ्यौँ भन्दाखेरि डिएनए हुन्छ, डिएनएमा पर्टिकुलर सेट अफ इन्स्ट्रक्सन हुन्छ एसीजीटी, यो चारवटा प्रोटिनको कम्बिनेसनबाट, अनि यो चारवटा प्रोटिनको कम्बिनेसनबाट एउटा पर्टिकुलर टाइपको मेसेज बनाउने हो, ठिक छ। अनि त्यो मेसेज के हुन्छ भन्दाखेरि हरेक सेलमा यी सबै मेसेजहरू सेम हुन्छ, खालि फरक के हुन्छ भन्दाखेरि एउटा पर्टिकुलर सेललाई एक्टिभ गराउनको लागि एउटा सेट अफ डिएनए मात्र एक्टिभ हुन्छ, अरू बाँकी सबै सुत्या हुन्छ, ठिक छ। जस्तै नर्भ सेलको लागि पर्टिकुलर सेट अफ त्यो डिएनए को इन्फर्मेसन एक्टिभ हुन्छ, अरू सबै सुत्छन्। मसल फाइबरको लागि सेम मतलब डिफ्रेन्ट टाइपको डिएनए को इन्स्ट्रक्सन सेट एक्टिभ हुन्छ, अरू इनएक्टिभ हुन्छ। त्यस्तै गरी अर्को इन्टेस्टाइनल सेलमा पनि अर्को टाइपको डिएनए सिक्वेन्स चाहिँ एक्टिभ हुन्छ, अरू चाहिँ इनएक्टिभ हुन्छ। यो कसरी गराउँछ भन्दाखेरि mRNA ट्रान्सक्रिप्सनको त्यो प्रोसेसबाट गराउँछ, फर विच नोबेल प्राइज 2024 इन फिजियोलोजी अर मेडिसिन वाज अवार्डेड। सो यही नै थियो नोबेल प्राइजको। अनि जस्तै यो एक्सपेरिमेन्ट चाहिँ केमा गरेको थियो भन्दाखेरि यो कहिले गरिएको थियो, अनि यो एक्सपेरिमेन्ट चाहिँ यो नोबेल प्राइज दिनको लागि कुन चाहिँ एक्सपेरिमेन्ट अथवा कुन चाहिँ डाटावाला उनीहरूलाई यसलाई ब्याक यो नोबेल प्राइजलाई ब्याक गर्ने कुन चाहिँ एक्सपेरिमेन्ट थियो त्यो छ। जस्तै लेट मी पुल आउट द स्क्रिन अगेन। एकदम ठिक छ, नै उता स्क्रिन भएन। So, यहाँ छ, research on a small worm leads to big breakthrough. Yes, right. In the late 1980s, Victor Ambros and Gary Ruvkun were postdoctoral fellows alongside in the laboratory of Robert Horvitz, who was awarded Nobel Prize in 2002, alongside Sidney Brenner and Solston. So, Victor र ग्यारी चाहिँ पोस्ट डक भनेको पिएचडी पछिको पोस्ट डकको एउटा पोजिसन हुन्छ त्यो फेलो थिए इन द ल्याबोरेटरी रबर्ट हबिजको चाहिँ रबर्ट हबिजको ल्याबमा वाज अवार्डेड नोबेल प्राइज इन 2002 अनि हबिजको ल्याबमा चाहिँ उनीहरूले एउटा 1 मिलिमिटर लङ राउन्ड वर्म त्यो राउन्ड वर्मको साइन्टिफिक नेम चाहिँ सी एलिगेन्स भन्ने साइन्टिफिक नेम हो अनि यसको यो साइजकोमा पनि स्पेसल टाइपको सेलहरू चाहिँ एक्जिस्ट गर्दा जस्तै नर्भ एन्ड मसल सेल विच आर फाउन्ड इन लार्ज एन्ड कम्प्लेक्स अर्गानिजम होइन सो यो चाहिँ यो अर्गानिजम के बन्यो भन्दाखेरि एउटा मोडल बन्यो टु स्टडी हाउ टिस्यु डेभलप एन्ड मेच्योर इन मल्टिसेलुलर अर्गानिजम अनि एम्ब्रोज र रुकुनले के गर्थ्यो भन्दाखेरि उनीहरू त्यसको जिनमा इन्ट्रेस्टेड थिए अनि त्यो जिन कुन केले कन्ट्रोल गर्छ एक्टिभेसन गर्नको लागि होइन त्यसको लागि उनीहरू इन्ट्रेस्टेड थिए अनि उनीहरूले चाहिँ दुईवटा म्युटेन्ट स्ट्रेन अफ वर्म्सलाई हेरे जस्तै लाइन 4 अर्को लाइन 14 होइन अनि उनीहरूले चाहिँ त्यो स्ट्रेनलाई किन चुज गरेको भन्दाखेरि उनीहरूले चाहिँ त्यो एक्टिभ त्यो जुन जेनेटिक प्रोग्राम हुन्छ नि त्यो डेभलपमेन्ट हुने फेजमा त्यो चाहिँ एक्टिभ हुने टाइमिङ चाहिँ डिफेक्ट थियो त्यो यो दुईटा स्ट्रेनमा सो उनीहरूले चाहिँ ठ्याक्क कुन चाहिँ म्युटेटेड जिन हो अनि त्यसलाई त्यसको फङ्सनिङ के हो त्यसलाई अन्डरस्ट्यान्डिङको लागि उनीहरूले चाहिँ के गरेका थिए भन्दाखेरि उनीहरूले यसैमा काम गरेका थिए So Ambros had previously shown that line 4 gene appeared to be a negative regulator of the line 14 gene. भनेको के भन्दाखेरि लाइन 4 को जिनले चाहिँ लाइन 14 को जिन को लाइन 14 को जिनलाई चाहिँ नेगेटिभली रेगुलेट गर्थ्यो रे है अनि त्यसपछि त्यसले गर्दाखेरि चाहिँ लाइन 14 एक्टिभिटी चाहिँ लाइन 14 को एक्टिभिटी जुन ब्लक भएको थियो त्यो चाहिँ पहिला अननोन थियो त्यो चाहिँ पहिला थाहा थिएन सो अनि त्यसपछि यो पोटेन्शियल रिलेसनसिप अनि यसको मिस्ट्रीलाई सल्भ गर्नको लागि चाहिँ उनीहरू चाहिँ इन्ट्रिग थिए अनि त्यसपछि तल यहाँ डायग्राम छ जस्तै यो सी एलिगेन्सको यो 1 मिलिमिटर साइजको एउटा अर्गानिजम हो त्यसपछि फिगर बीमा लाइन 4 र लाइन 14 म्युटेन्ट्स यु क्यान सी नर्मल्ली यो कुन चाहिँ हो सी एलिगेन्स अर्गानिजम लाइन 4 म्युटेन्टमा अनि लाइन 14 म्युटेन्टमा अनि त्यसपछि यो सी एलिगेन्सको जिनोम है अनि त्यसपछि उनीहरूको कल भयो उनीहरूको बिच कल भयो बिकज दे वर डुइङ एक्सपेरिमेन्ट अनि त्यसपछि उनीहरूले चाहिँ के गरे भन्दाखेरि लाइन 4 र लाइन 14 को माइक्रोआरएनएमा चाहिँ उनीहरूले चाहिँ के भेटाए दे फाउन्ड अ डिफरेन्ट सिक्वेन्स जस्तै लाइन 14 को पर्टिकुलर सिक्वेन्स र लाइन 4 को पर्टिकुलर सिक्वेन्स के भयो दे फाउन्ड डिफरेन्ट सो Ambros discovered that line 4 gene encoded a tiny RNA, microRNA, that did not code for a protein. Ruvkun cloned the line 14 gene and the two scientists realized that line 4 microRNA sequence matched a complementary sequence in line 4 mRNA. So, के भयो भन्दाखेरि उनीहरूले चाहिँ जुन म्युटेसन थियो नि त्यो म्युटेसनको चाहिँ रुकुनले क्लोन गर्यो लाइन 14 जिनमा अनि त्यसपछि उनीहरूले के गर्यो भन्दा लाइन 4 को माइक्रोआरएनए सिक्वेन्स चाहिँ कम्प्लिमेन्ट्री रहेछ भनेको जस्तै कम्प्लिमेन्ट्री भनेको के हुन्छ भन्दाखेरि अब कम्प्युटरमा हेर्ने भने चाहिँ 0 को कम्प्लिमेन्ट 1 हुन्छ ठ्याक्क फ्लिप हुन्छ ठ्याक्क फ्लिप खान्छ सो यही नै थियो लाइन 4 र लाइन 14 को अनि त्यसपछि तर यो आर्टिकलमा पनि छ फर्दर डिटेल तर आज हामी अब अहिले हामी धेरै नजाउँ यसमा म यो लिङ्क चाहिँ च्याटमा हालिदिन्छु सो द्याट यु क्यान स्टडी yourself and well, enjoy. 7 minutes यो त सुरु हुनु पर्ने होला नोबेल प्राइज कमिटीबाट नै लाइभ मेसेज के आएको छैन 7 December yes आज 7 December at 6:30 yes we are waiting Nobel Prize oh सुरु भयो alright guys Nobel Prize committee को message सुरु भयो we have live transmission and let's begin so be aware that Nobel Prize will the entire the academic भित्र you have to dress well you have to be entire etiquette नै एउटा different type को छ and you have to be very strict about it so हामी यहाँ पनि in live यसमा पनि हामी will try to maintain certain etiquette so that disturbance नहोस् right so let's hope it happens No, I didn't do it.
Guests, there are still many people waiting in line outside hoping to get in. We ask you to please take your seats so that we can now fill remaining empty seats. Thank you.
See, भनेको थिएँ नि त्यो एकपटक सेसन सुरु हुँदाखेरि एब्सोल्युट साइलेन्स भयो नि यो सदा राइट यस्तो खालको एटिक्वेट त्यहाँ पुरै मेन्टेन भइसकेको हुन्छ उनीहरू त्यहाँ जति पनि इन्भाइटेड त्यहाँ गेस्टहरू मोस्ट अफ देम आर इन्भाइटेड अनि प्रेसहरूलाई पनि इन्भाइट गरेको हुन्छ एन्ड दे हेभ बिन गिभन स्ट्रिक्ट इन्स्ट्रक्सन्स कि तिमीले यस्तो यस्तो गर्न पाउने छैनौ भनेर स्ट्रिक्टली मेन्टेन हुन्छ सो note that.
We would like you to pay attention to the following fire safety information. In the event of an evacuation, you will hear a recorded voice alarm message. Follow the instructions and evacuate through the nearest exit, marked with a green emergency exit sign. Fire wardens in yellow vests will direct you to the assembly points where you will get further information. Thank you for your attention.
Okay, host has entered to the stage. So अब चाहिँ नोबेल प्राइजको लेक्चरको लागि अब सायद केही सेरेमनी हुन्छ अनि देन लेक्चर चाहिँ सुरु हुन्छ. ओके, एडिक्वेट.
Distinguished Nobel laureates, honored guests. It's a great pleasure and privilege to welcome you all to the 2024 Nobel Lecture in Physiology or Medicine. My name is Annika Östman Wernersson, and I'm the president of Karolinska Institutet. During the Nobel Week, we pay tribute to contributions made by exceptional individuals whose discoveries, in the words of Alfred Nobel, have conferred the greatest benefit to humankind. These are words that remind us of the profound impact that science has on our lives. Today, I'm honored to extend my warmest congratulations on behalf of Karolinska Institutet to our new Nobel laureates, Professors Victor Ambros and Gary Ruvkun. Their groundbreaking discovery of microRNA and its role in regulating gene activity is a hugely important breakthrough. This year's Nobel Prize in Physiology or Medicine highlights the importance of curiosity-driven research. The discovery of fundamental new mechanisms of gene regulation would probably not have been achieved if goals for the research had been predetermined. I very much look forward to learning more about your findings and how they affect our deeper understanding of the biology of our bodies. Now, I will give the floor to Professor Anna Widell, member of the Nobel Assembly, who will further introduce this year's laureates. Thank you.
Esteemed Nobel laureates, dear colleagues and friends, dear students, on behalf of the Nobel Assembly, I welcome you all to the Nobel Lectures 2024. I have the honor and great pleasure to introduce Dr. Victor Ambros and Dr. Gary Ruvkun, who have been awarded this year's Nobel Prize in Physiology or Medicine for the discovery of microRNA and its role in post-transcriptional gene regulation. The flow of information is central to life. How different organisms perform the critical functions they need to develop, thrive, reproduce, all depend on a basic set of instructions that resides deep inside our cells in the form of genes that are made of DNA containing the code of life. How then is the flow of information controlled such that each specific cell type ends up with exactly the right combination of proteins at each specific time, despite the fact that the basic instructions are the same for all? Many parts of this flow of information have been understood thanks to previous groundbreaking discoveries. The central dogma of molecular biology states that DNA is transcribed into messenger RNAs that carry information for translation into proteins, which perform the specific functions needed for the organism. The central dogma of molecular biology is classic textbook knowledge, taught in schools around the world to students from a relatively young age. We all know it. This year's laureates, Victor Ambros and Gary Ruvkun, have conceptually changed the picture by adding a completely new level of understanding to this classic problem. They both studied heterochronic mutants of the tiny worm C. elegans. These are strains of worm that display changes in timing of development. The focus was thus on peculiar-looking worms, anticipating that they could provide fundamental biological insights. Two participants then demonstrated that deletions in the gene's untranslated region affected protein abundance in some mysterious way. Ambros, on the other hand, embarked on the huge task of cloning the first gene and made the surprising discovery that it encoded a tiny RNA without protein coding capacity. Dr. Ambros and Ruvkun compared their results, and as we will hear, this resulted in a dramatic discovery moment when they realized that the tiny RNA was capable of astonishing things never heard of before. Dr. Ruvkun later made an additional dramatic discovery leading up to the realization that tiny RNAs are widely spread all through the animal kingdom, defining the class of microRNAs with thousands present in the human genome. Victor Ambros and Gary Ruvkun thus unraveled a completely unexpected mechanism for how flow of information is regulated in our cells. MicroRNAs determine which of all messenger RNAs are translated into proteins, thus exerting a fine-tuning control station that operates differently from all previously known modes of gene regulation. By working in networks, they allow for combinatorial effects, enabling systems-level actions. In complex life, myriads of complicated biological processes are active simultaneously. All of these have to be precisely regulated without interfering with each other, while also compensating for changing environmental conditions. How is all this possible? Shouldn't it all be chaos? The discovery of microRNAs with their systems-level mode of action does make it a bit more conceivable. We are going to hear an exciting story by two remarkable individuals. Both showed a strong interest in science already from a very early age. As boys, they were fascinated by astronomy, growing up during the early days of space travels. Today, they are passionate scientists who have devoted large parts of their lives to curiosity-driven research, characterized by creativity, open-mindedness, technical ingenuity, strong perseverance, and data sharing, leading up to a fundamental discovery with great benefit to humankind. We now know that microRNAs play important roles in most physiological processes and are involved in a range of human diseases. On behalf of the Nobel Assembly, it is now my great pleasure to introduce the Nobel laureate Victor Ambros. Victor Ambros was born in Hanover, New Hampshire, USA. In 1975, he received a Bachelor of Science with a major in biology from the Massachusetts Institute of Technology. In 1979, he received his PhD from the same institute after studying poliovirus genome structure and replication with David Baltimore. Dr. Ambros then joined the laboratory of Robert Horvitz at MIT as his first postdoctoral fellow, remaining there until 1984. He then took a faculty position at Harvard University. Between 1992 and 2007, Dr. Ambros was a professor of genetics at Dartmouth Medical School. In 2008, he joined the faculty at the University of Massachusetts Medical School. Dr. Ambros is now Silverman Professor of Natural Sciences at the University of Massachusetts Chan Medical School, Worcester, Massachusetts, USA. Dr. Ambros We are very much looking forward to your Nobel lecture entitled A Marvelous Unfolding Story of MicroRNAs. Please join me in welcoming Dr. Ambros onto the stage.
All right, Michael Ambros. Okay, is that our lecture students? Okay.
Thank you so much.
So pay attention.
Thank you. Thanks to everybody for being here. I hear it was standing in line was a pain in the neck, but I'm glad you were able to come. I want to thank everybody who's out there who's ever contributed in any way at all to me being here in this moment. And I'll try to mention some of those people as I go along. I bring you greetings from Worcester, Massachusetts. I'm going to tell you a story that basically is about a story of a paper that we published in 1993. So this story is about more than 30 years old. And the individuals who did the work are in this room. That's Rosalind Lee and Rhonda Feinbaum. So this talk is dedicated to them. First, how did I get here? All right, as you heard, I was born in Vermont. I think I started out as a scientist. I don't remember not feeling like a scientist, and that's my dad who encouraged me along the way. I like to show this because it shows that I was a good student. I got A's and B's, but also it points to the signature of one of my favorite teachers in the 4th grade and the 6th grade, Wally Vane. And also it allows you to see the marvelous signature of my dad. He wrote everything like that, including letters, checks. People would, he'd write a check to people and they would not cash it for a while because they wanted to show all their friends. So I was an amateur astronomer, took some pictures, and this is my first published scientific result. In Sky and Telescope magazine, 1970, we had visited Virginia to see the total eclipse of the sun, and I observed shadow bands, which are a phenomenon that sometimes is visible during totality or just before and after totality. And here's the thing right here. Victor Ambros found the bands visible for over 4 minutes before totality and almost 3 minutes afterward. That's in a real magazine, my name. And it says his station was near Chincoteague, Virginia. This is my station. I think that's the first time I felt like a real scientist right there. So my path, as you heard, started at MIT, finished in 75. I was influenced a lot by people around me. Here we are throwing a piano off the roof of my dorm. And that was the first of what became an annual piano drop at MIT that still happens today. That's me. I was actually influenced a lot by other students. So for example, the Struhl brothers were people who I got to know, and they were doing research in a lab, and I realized, wow, people can do research in labs, and they were so enthusiastic about biology that I got converted to biology. Ed Gruberg was a really important mentor of mine, taught me how to read literature and write a paper. I learned so much from David Baltimore, I can't possibly start to enumerate here today. Ditto, Bob Horvitz, although I'll try to mention a few things that Bob taught us. And then I had these faculty positions. I was recruited to UMass by Craig Mello, who used to be, before that, my PhD student. And so I have this Nobel connection that's, I think, fairly extraordinary. I've worked with 3 Nobel laureates. So I'm here to tell you this is an improbable life. So the story of this project begins in 1980, Bob Horvitz's lab. And shortly before then, and then soon after, Bob and John Solston put together different parts of the development of this worm into a complete cell lineage. And the way they did this was they sat in a room looking through a microscope, taking notes, watching cells divide in living animals, right, and drawing them as they divided and moved around, underwent programmed cell death, migrated here or there, and differentiated. And they did that enough so they were able to draw this lineage and say that it's invariant. They never the animals in wild type never make a mistake. They do this lineage with this timing of cell divisions and these fates associated with these patterns, from animal to animal. So that was part of why the system was attractive to people, because it seems you could follow, analyze development on a cellular level in real time. I think also in retrospect, this invariance of the lineage offered to us a way of studying how developing animals are able to robustly develop given the stresses of everyday life. These animals live in the dirt. They have to eat whatever bugs they can find, bacteria. They live over a wide range of temperature. But the cell lineage development is invariant regardless. So the big question was, how is development encoded in the genome of an animal? Bob is using a genetic approach, which is the unbiased approach. You find a mutant, and then you follow that mutant and find out what it can teach you about the system. It's not based on presupposed ideas about how things work. So the model organism premise is pretty cool, right? The worm is a tiny person in disguise. And so that means if you study developmental mechanisms in one animal, it'll teach you about other animals. And the rationale for this is basically that we have a common ancestor. That's pardon my fanciful bilaterian ancestor, but the idea is that that animal had an endowment of gene regulatory network machinery that possessed latently everything for all the animals after that. And we now know from genome sequences, estimates vary, about 5,000 genes that are completely conserved. And it's that genetic toolkit and how it's adapted and how regulatory interactions and mechanisms are brought into play are underlying, the diversity and complexity of animals. So the genetic approach is you've got a mutant and you've got a phenotype, right? So you figure there must be a mutation, and there must be a gene there. And you can verify that, as you all know, by Mendelian crosses. Okay, so there's a gene that's going to be in the DNA, so you got to find the DNA, figure out a gene product, learn what you can about potential biochemical activity, and ultimately you can relate that to your phenotype and how the mutation changes the gene, and make some inferences about normal function of the gene product, right? And in the 80s, this was a lot of work, right? Starting from a mutation and a phenotype, it could take easily 5 to 6 person-years to even, as we say, clone the gene so that you have the DNA and the sequence. So how do you choose what mutants to follow? And Bob recommended that we should follow these developmental timing mutants. And they're very interesting. You've heard about them. One is this LIN4, loss of function mutant, which I'll tell you about first. The 0 there means the mutation is, we think, knocking out the activity of that gene. And it turns out the mutation we worked with was indeed knocking out the activity of LIN4. So that's what happens to the animal when it doesn't have LIN4. It ends up developing in a pretty crazy way. Many of the cell lineages in the animal are repeating what normally would happen only in the first larval stage. So you can see, the blue circle and the pink circle, those are different patterns of cell division and cell fate. The blue thing is basically a neuroblast is produced there. And in the LIN4 mutant, things that happen later don't happen at all, and throughout numerous lineages, you see this happening. And this was worked out by Marty Chalfie, working with Bob and John Sulston. So when I came to the lab, this was known. It was a really interesting mutant, LIN4. And it had, so Bob taught us to make simple models, right? Okay, the phenotype is you repeat the first larval stage, you don't make the second larval stage fate, so it must be that the normal function of LIN4 is to specify the second larval fates and or prevent first larval stage fates. So when you remove the gene, look what happens. You repeat the first larval stage phase. These we called heterochronic mutants as a nod to the concept of heterochrony in evolution, where, for example, ancestral juvenile traits are maintained in a descendant generation. And so, this mutant is basically like that. It's retaining ancestor juvenile traits. So the opposite kind of phenotype, this was actually identified by Chip Ferguson in Bob's lab. These mutants, the Lin14 loss of function, do not do the fates that normally would happen in the first larval stage, and they skip right to the second larval stage. And this happens in many different cell lineages, so it's a sort of a global kind of transformation in cell fate to a later cell fate. So again, our simple model is that Lin14 could specify L1 fates and or inhibit L2 fates. So these two opposite phenotypes leave us with two nice little models, right? And Bob taught us to make regulatory pathway models. And so there's two alternatives, right? Lin14 might inhibit Lin4, which is specifying L2 phase. Or Lin4 might inhibit Lin14, you know, and that will control transition from L2 to L3 phase. So which is it? And so, all right, get ready. This is the simplest experiment in the world, right? You make a double mutant, okay? And what happens? A Lin4, Lin14 double mutant looks just like a Lin14 null, single mutant. So Lin4 doesn't matter if Lin14 is missing. So the idea is that's the pathway that must be the case. And so, you know, considering this, it's interesting to contemplate that, okay, what's going on in these mutants is cells are being transformed sort of in their cell fate in a temporal direction. And this implied that in these lineages in the wild type, that cells were being produced, stem cells essentially were being produced with similar developmental potentials. So the cell in this lineage in the first larval stage has both potentials for doing that L1 developmental program or that L2. Ditto for that stem cell produced at a later stage in that lineage. And what happens in the mutants is the fates get transformed such that what normally gets whatever option gets normally chosen at that stage is not chosen properly. And according to our nice little model at the bottom, that would be accomplished by Lin14 being present and then absent under the repression of Lin4. So this was interesting and justified putting a lot of effort into maybe cloning the Lin14 and Lin4 genes. And Bob recommended that Gary and I take this project with us to our respective labs, and sort of informally we split it up. My lab would concentrate on Lin4 and Gary's would work up Lin14. And then we were starting in each case with mutants, okay? I wanted to remind you of that. In our case, one mutation, Lin4, E912. You'll get used to that little number in a bit. And Gary's going to tell you about some really fascinating Lin14 gain-of-function mutations as well. All right, so we're getting started. What do we know? We have a genetic map, okay, recombination map. Lin4 is between blister 2 and dumpy 10. Our mutation is E912. This one, by the way, had an interesting provenance. This mutation was generated in the Brenner lab by feeding worms P32 labeled bacteria. And when they ate the radioactive bacteria, the P32 would degenerate and break their genomes and make mutants, and somebody screened through those populations of animals that were eating P32 bacteria for mutants, and E912 came out. So when we started this project, the whole project was based on a mutation that happened to arise by accident in the Brenner lab as a result of a P32 molecule, atom degenerating and shooting out whatever it does, a gamma ray that broke the DNA, okay? So that's how careers start. We were incredibly supported by the Medical Research Council MRC project led by John Solson and Alan Coulson. And about the time we started, here's 1988, this is a report in the Worm Breeders Gazette. I don't know how many of you have been around long enough to have subscribed to the Worm Breeders Gazette, but I happen to know there's a few people in this room who have. This was an amazing thing where it was, you know, before the internet, basically, people would post their unpublished results. And Alan and John would periodically post the latest physical map. They were working out the overlaps between all the clones that they had collected, DNA clones. They would share clones with us, of course, send us advice. Here's I found a couple of letters from Alan and John. And this is, Rosalind worked with this specific map right here. You can see each line is a cloned piece of DNA. Cosmids or yeast artificial chromosomes, the longer ones, they had worked out these overlaps by a high throughput fingerprinting method. But they didn't know where the genes were, right? So they were working in parallel then with labs like ours and dozens of other labs to put the genetic map in register with the physical map. And of course, this was a prelude to what became the C. elegans genome sequence. So Dumpy 10, for example, it's a collagen. Somebody assigned it there. Look, it's just written in hand. So now we know, okay, so Lin4 must be off to the left, right, somewhere. We don't know how far. So Rosalind's first task was to identify clones from these collections here that overlap strain-specific DNA variations, or RFLPs. Okay, so hang on to your hats. If you're a geneticist, you'll like this. Otherwise, whatever. These are beautiful things. We wanted to find a piece of DNA that we could have in our hand and that we could say, this piece of DNA has the following phenotype. It recognizes a piece of the genome that differs between two strains. And we can assay that difference by a southern blot. You can look up southern blot, young folks, on Wikipedia, if you want. And you can map it now. It's a genetic trait. So you make a heterozygote between one strain that doesn't have this polymorphism and another strain that does. It's RW7000 strain. It has lots and lots of transposon insertions across the genome. So this allows you to detect these polymorphisms pretty easy. And you take a probe and probe it to your southern blot of these recombinants. You pick a bunch of recombinants that are all dumping on Lins, and then collect those and analyze them by southern blotting and ask which ones retained the MAP12 trait or not. And that maps that particular piece of DNA to a position genetically near Lin4, right? And much closer than Dumpy 10. That's Rosalind's toehold in the genome. So using the MRC, Alan and John overlapping clones, she marched along and basically tested them as she went for whether or not the clones could, on southern blot analysis, detect differences between the E912 mutation and wild type. So think about this for a minute. If E912 had been a point mutation, right, there's probably not a chance in the world, really, unless that point mutation affected a restriction site. All right, so now we're getting gory, and if you're a molecular biologist, you can appreciate that. So the chance of finding a Lin4 E912 mutation was risky, right? But she found it. And it turns out that, so for example, here's a blot with two different enzymes digesting DNA from Lin4 E912 versus wild type, and you can see the difference, right? The wild type is much brighter and it's different in size. That's because a lot of that piece of DNA that she's using as a clone is deleted. And so, by the way, for these experiments, Rosalind benefited a lot from some new technology. This is like late 80s hot technology called pulse field gel electrophoresis that enabled you to take these yeast artificial chromosome clones that Alan Coulson and John Solson and Bob Waterston were producing and mapping, and you could purify them using this. And she used this. This is an MJ Research controller. This was the first product of what became MJ Research, which some of you may recognize as a major supplier of thermocyclers for a number of decades. And this was made by Mike Finney and his brother John. Mike Finney was a graduate student, I think, in Bob Horowitz's lab at the time. So Rosalind took up the task now of sequencing this piece of DNA that was detecting the E902 mutation. But here's a problem at this point. Okay, so it's a big deletion that allowed us to see the lesion, but it doesn't really tell us with much resolution where Lin4 is. So we needed a point mutation, and Rana took up the task to conduct a series of high-throughput, high-volume crosses where the males are mutagenized and then crossed to a strain that is E912, right? And it's marked with a dumpy so that you can tell self from cross. And so the cross progeny will mostly be normal because E912 and the dumpy are recessive. The goal is to find a rare animal that looks like Lin4 E912, not because it's homozygous for E912, but it's heterozygous for E912 plus a new allele that knocks out Lin4, right? And Lin4 seemed to be a small target, right? Because these were very rare, unexpectedly rare. but she got one, and it's a point mutation, and it's called MA161. So that told us that the Lin4 gene product must traverse that part of the sequence, right? What about the boundaries of Lin4? So the strategy that Rhonda and Rosalind used was injecting clone DNA into C. elegans to generate transgenic worms. This methodology was worked out, by a number of people, including Craig Mello optimized it. It was magically, so to speak, you can just inject into the worm DNA, and at quite high frequency, the progeny will now have that DNA, and it's inheritable. So you can ask, does the DNA that I inject confer wild-type Lin4 activity, meaning will it rescue the phenotypes of those Lin4 E912 mutants? And that clone did, a smaller clone did, a smaller clone rescued, an even smaller clone, and finally, 700 base pairs clone rescued Lin4. So this is a really small gene, and that started to pique our interest because even as a protein gene, this is going to be a small protein. So we felt we were onto something interesting. So the Lin4 gene has to be in there. We want to find the transcripts. So those sequences are used to probe, as probes, try to find the transcripts through various hybridization strategies. And Rhonda went on maternity leave, I think, and so I did for a little while. I actually did some stuff, and doing these ribonucleotide protections. And here you have a radioactive probe. You hybridize the total RNA of wild type or your E912 mutant, and you ask what size of a piece of the probe gets protected from ribonuclease because it's formed double-stranded. And we got really excited because it's 55 nucleotides. So I'm telling you, wow, we knew we had something unusual. The gene is small, and the transcript is only 55 nucleotides, or maybe a little bit more than that, depending on interpreting the probes here. So we knew it was small. But then, so I presented these data at a floor meeting, a research in progress, and our friend Margaret Barron said, "What's that at the bottom of the gel?" And I said, schmutz, I don't know. And she says, well, maybe you should look at a shorter exposure. And sure enough, the major transcript, which is present in the wild type, present in the rescued strain, absent in E912, absent in the control, is really small. So the major transcript is small, about 22 nucleotides. And Rhonda now really nailed this down with northern blots and S1 nuclease protection and primer extension experiments. So there we were. Now we definitely had something unusual. There's the MA161 mutation in one of the nucleotides that's in that major transcript. The minor transcript, longer, folds into a hairpin. We now know that's the precursor that gets processed out by Dicer and captured by Argonaut. So, and there's MA161. So the thing is, we think, okay, this thing has a very small transcript. It's really unusual. And we're going to report to the world that that Lin4 does not encode a protein, right? And I think that I was annoying, perhaps, to Rhonda and Rosalind because I thought, okay, we got to prove it. We got to prove there's no protein whatsoever. I mean, there are some start codons, but of course there's some stop codons too. So Rhonda and Rosalind did some mutagenesis, inserting out of frame, substituting in a stop, all those constructs rescued, so those putative upstream orphs can't be real. And then they did evolutionary mutagenesis. Rosalind cloned Lin4 from 3 other nematode species. These species are maybe 20 million or so years different from C. elegans from each other. So you're letting evolution do the mutagenesis. And importantly, all those clones from the other species rescued C. elegans Lin4. So they have Lin4 function, but if you look through the sequences, you basically cannot find a conserved protein. So we nailed it. There's no protein produced from that gene. So in June 1992, Gary and I got together and compared sequences because we had the small RNA sequence. He had sequence from Lin14 3' UTR, and we matched that up. We got on the telephone. I don't know how many of you recognize that as a telephone. And it's a landline. And there was this complementarity between these 3' UTR elements in Lin14, and importantly, Gary's lab has shown that those elements were conserved in multiple nematode species, just like we had shown that the Lin4 was conserved in sequence. So the complementarity was conserved. So in that moment, we knew the mechanism. It's really wonderful, because it's rarely that you can kind of inspect data and know the mechanism. So that was definitely fun. So we published those papers. Nowadays, by the way, we know that Lin4 is a little bit shorter than what we had it. We're being generous, given the vagaries of the methods at the time. Okay, so what did we do then? So from 1993 to 2000, our lab was doing some heterochronic stuff. The pathway was a little bit crude in the beginning, about that time. We knew that there was Lin4, Lin14, there was Lin28 was involved, and these were controlling early larval fate transitions. The Lin29 was controlling later transitions, and Rugby cloned Lin29, showed that it was actually a conserved zinc finger protein. And Eric Moss, working with Rosalind, cloned Lin28. That was also a conserved protein, in this case an RNA binding protein apparently. Also, they found it was targeted by Lin4. So this Lin4 mechanism, whatever it is portable to another gene, as long as you have sites in the 3' UTR for Lin4. And then Rhonda did some nice experiments showing that Lin4, in fact, acts as a switch. Phil Olson did some interesting work on the mechanism of Lin4 repression of Lin14. I'll mention that in a few moments. And then working with the Ruvkun lab, found that Lin41 is a conserved protein and it's targeted by LET7. So there were 3 conserved proteins in the pathway. So the heterochronic pathway had, gained a lot of cachet at this point. But, up until 98, though, Lin4 was the only microRNA, and even at 99, Lin4 and LET7 were the only microRNAs, and they're only in C. elegans, right? So, but what was going on in parallel was a really interesting consilience of microRNA and RNAi phenomena. So Fire and Mello discovered RNAi and C. elegans, and Hamilton and Balcombe showed right about the same time that in plants, RNAi was associated with a blooming of small RNAs of about the same size as microRNAs. So it really looked like RNAi and microRNAs are maybe mechanistically related. This was really nailed down by the Mello lab working with Ruvkun, showing that Argonaut proteins are central to RNAi and microRNAs, right? So this really told us that, look, maybe microRNAs, Lin4 and LET7, are evolutionary adaptations of RNAi for gene regulation, right? And let me remind you, RNAi is generally, a viral defense mechanism where double-stranded RNA is detected. Foreign double-stranded RNA usually is detected. Viral replication intermediates, for example. Dicer cleaves that up into small RNAs. They go into AGO. One strand and it goes into AGO and it destroys the invading RNA. So microRNAs are double-stranded RNAs that are processed by Dicer, but the double-stranded RNA comes from the genome. It's in a gene that's evolved to regulate other genes, right? So RNAi is ancient, so microRNAs. LET7 is conserved across all the animals. This paper, when it came out in 2000, was a real watershed. And personally, I didn't even know these results. These guys had kept it secret, right, for a maximum drama. So, when I opened Nature and read this, I thought, holy cow, wow, So microRNAs, LET7 is ancient, derived from RNAi. The bilaterian ancestor had LET7. Look, it's been 400 million years. It's had plenty of time. It's just, life has to evolve other microRNAs besides LET7. And we just haven't found them, right? Because they didn't present themselves to us by mutation, right? Okay, so in a model organism. So Rosalind and I got excited, and we taught ourselves all kinds of new stuff, like making cDNA libraries from small RNAs, growing up these, making libraries. We just hadn't done this stuff before. We found finding somebody who could sequence little short inserts without charging a fortune, or as the Whitehead wanted to do, be co-authors and charge a fortune. So then we could match them to the genome, and sure enough, the genome where they matched could be folded into a hairpin, and so they look just like Lin4 and LET7, but they're different sequences, and many of them are conserved in other organisms, including flies and humans, just by sequence match. So we thought, well, we got to write this up. That's going to be cool. But then one day I get an email from the editor of Science and says, can you review this paper? Here's the abstract, and the abstract from the Tushel lab, and it says, we've cloned many new microRNAs, from humans, and then they matched Drosophila and C. elegans and stuff, and so there's way more microRNAs out there than Lin4 and LUT7. So it's like exactly what we wanted to say. But so I said, no, I can't review it because we have similar results, and is it okay if we send our manuscript to be, considered at the same time? And he said, very kindly, he said, yes, but get it to us by the end of the week. And there's another paper from the Bartel lab, So I'm telling you, by the skin of our teeth, we got this paper co-submitted and co-published side by side with these other papers. And together, the scope of these papers varies, and ours was focused on C. elegans, but basically the message from these three is there's lots of microRNAs, new ones, and a lot of them are conserved, like LUT7 in all these animals, in flies, worms, and humans. So lots of other papers followed from that and sort of filled things in, and including showing that Lemire 100 is in Nigeria, meaning that it's even more ancient than the bilaterian microRNAs. And nowadays we know of about 25 bilaterian seed families. And the way we refer to them as seed families is because position 2 through 8 of the microRNA, which is the part that nucleates binding to targets, is conserved. That's the part that gets fixed in evolution once a microRNA evolves. So, and by the way, there's other microRNAs in sponges, fungi, and plants, but they're probably evolved independently from RNAi. So there's maybe, 500-600 genes, microRNAs in humans, we now know, 100 or more. Seed families, a couple hundred, 100 to a couple hundred. How do microRNAs recognize their targets? Here's an early study that really kind of told us. This is a Brennecke paper showing that for these microRNAs and these targets, you can see base pairing predicted between the seed, and in some cases in the middle, seed only, and in other cases, seed plus 3' sequences. What's shown is, I think, 5 or 6 species of insect. So this conservation of the base pairing or the complementarity is used pretty routinely to apply confidence to the fact to the idea that these interactions are functionally real. So we search for potential targets of a microRNA computationally using the seed sequence, essentially. Now, how do microRNAs repress the target? So the key thing here is you notice that the microRNAs in animals don't perfectly match their target like siRNAs do in RNAi. That activates the slicer activity of Argonaut, and the RNA gets destroyed. It appears that evolution has contrived to allow animals flexibility in how they use their microRNAs. Most of the targets of all the microRNAs, essentially, are mismatched at least position 9 and 10 of the microRNA. So this gives the situation some flexibility. There's no slicing, no rapid destruction of the mRNA, and this allows what's called a programmable repression by other means. And we know that Argonaut recruits a variety of proteins, assembles complexes that apparently have specialized or programmable outputs to either repress translation in some fashion or to cause the mRNA to be destabilized, often through deadenylation, and then decapping, and then destruction. So there's lots of examples, and I want to emphasize, of microRNAs repressing targets without destabilizing the mRNA. So this is a early example from Brennecke et al. 2003, which in my opinion is actually still the best paper in the field of microRNAs. It's really amazing. Done by 2 graduate students in about 13 months, and published it in Cell. The Bantam microRNA represses HID protein production, and that regulates, is involved in apoptosis of the fly. So you can see here, these are wing primordia that they dissected out of the caterpillar. And the left panel is the protein detected by an antibody, and then the right panel is a primordial way they've detected the mRNA in this fashion. And you'll notice if you're a fly geneticist, you know they used this beautiful and elegant style of experimentation where they expressed hid in a stripe down the middle of the primordium, right? These are in flies that are not making the bantam, not producing the bantam. In flies producing bantam in this wing disc, you see the protein is essentially erased, but the mRNA is still there. So somehow the hid bantam interaction ends up in translational repression of the target. Here's another example. This is familiar Lin4, Lin14. Phil Olson in the lab did these studies. If you take L1 larvae and grind them up and run them on a sucrose gradient, you can stretch out their ribosome-containing complexes, and down near the, as you go to the right on this graph, these are mRNA complexes with more and more ribosomes on them. The Lin14 mRNA, detected here by northern blot, you'll notice lots of it is associated with polyribosomes. This is in the L1 when Lin4 is not present, lots of Lin14 proteins being made. In the L2, when very little Lin14 protein is being made, surprisingly the Lin4 message is still associated with polyribosomes. So this suggests some kind of mechanism where the translational repression is occurring in association with polyribosomes, perhaps freezing them in some fashion. These conclusions were supported by the Andy Fires lab working with 21st century technology. So this partial seed match, the seed match configuration with some 3' pairing allows you to search computationally for targets. And each microRNA tends to come up, prove to be predicted to regulate multiple targets. And so here's where I want to make a really cool point, not a cool point, but an important point that we don't really know the answer to. Okay, this target scan program is a conservative one. It identifies targets without a lot of false positives. But what's fascinating is that, for example, C. elegans Let7 has about 134 predicted targets, but actually the Ruvkun lab showed by genetics that the vast majority, or most of the Let7 loss of function phenotype is due to de-repression of one protein, Lin41. So one of the interesting open questions in this field is, for microRNAs, like, what's with all the targets? So I'll just quickly do this. In this study with Chen and Rajewski did was to basically, let's compare 3 species, flies, worms, and humans, for how many microRNAs they have in common. There's about 20 conserved seed families, and for about the 5,000 genes that are conserved across these 3 organisms, there's a very small minority of those genes that happen to be targeted by the same microRNA in all 3 species. So there's quite substantial evolutionary fluidity in targeting, and that's thought to reflect perhaps an adaptive role for gain and loss of microRNA targets. Now, at the same time, there are deeply conserved targets of microRNAs. And one kind is this Let7 Lin28 little epigenetic circuit where the RNA binding protein Lin28 directly regulates Let7 and Let7 directly regulates Lin28. And this controls pluripotency versus differentiation in many, many contexts, even in the same animal. So it's, in C. elegans, it's employed in our heterochronic pathway in this fashion. So one mode of action of microRNAs is developmental switches, like Lin4 and Lin4 on Lin14, Let7 on Lin28 or Lin41. Another interesting mode is related to switches. It's in this example, these neuronal microRNAs can be sufficient to reprogram directly fibroblasts into neurons. Another mode of action of microRNAs is coordinating suites of genes. And in this case, MIR1 is regulating multiple components of a multi-protein complex. And this turns out to be important for muscle physiology. Another role that's been shown in a number of contexts is microRNAs are involved in gene regulatory networks where they basically buffer the network output from noisy input. And noisy input is an inevitable sort of trait, let's say, of transcriptional regulation, for example. Okay, one of the ways I love these experiments where people ask, well, what do microRNAs do? So let's just make a fish without any microRNAs. And I don't want to go into detail, but it's a beautiful experiment where Antonio Giraldez, working with Alex Shear, made these fish that have no microRNA. What happens? They make a fish, okay? So you don't need microRNAs to make a fish. But the fish are simpler than they normally would be. They lack a lot of cell types and a lot of morphological features that normal fish have. They're also very stress sensitive. So, the take home is microRNAs are good for promoting developmental cell complexity, and conferring physiological and developmental robustness. Here's another example from flies. Let's make some flies without microRNAs. And so the Carthew lab did this. Not surprising, an AGO1 mutant, the microRNA Argonaut, those mutants fail in development. That's if they're developing normally. If you contrive to slow their metabolism by removing insulin signaling, so they develop very, very slow, at very, very slow metabolic rate, they can make flies. And similar results with if you're an aficionado in clones in the eye. So the idea is that they propose a really, I think, an interesting idea that microRNAs enabled rapid high energy growth and development, which would be an obvious adaptively advantageous innovation. All right, so they promote organismal complexity and stress-resilient living. Now, of course, stress resilience is intricately related to disease, right? And so this brings up the subject of, okay, what do microRNAs do in human disease? And I don't have any time here to cover the vast literature in this area. I just want to tell you one story that has been important to us because it's enabled us to get involved in our lab in a way of shifting from purely C. elegans, developmental genetics to a kind of partially C. elegans developmental genetics, but with some human context involved. And so I want to tell you about these human Argonaut mutations in neurodevelopmental disorder. And so these are mutations that occur spontaneously in people. They're not present in the mom or the dad, but the child has a neurodevelopmental disorder, delayed. It's many, many different kind of let's say indications, varies from child to child and so forth. These were striking because these are deletions of multiple Argonaut genes. So Argonaut 1, 3, and 4, AGO1, 3, 4 in humans are clustered together. These are microRNA Argonauts. There's one more microRNA Argonaut, AGO2 elsewhere. These mutations are knocking out multiple Argonauts, but they never found individuals that carried a knockout of a single Argonaut gene. Okay, so that suggested that to this indication, neurodevelopmental disorder, required that multiple Argonauts be compromised somehow. Okay, but there's the most prevalent mutations are point mutations, but these are all single amino acid changes in, as it turns out, evolutionarily conserved amino acids in AGO1 or AGO2, and now there's AGO3 mutations have arisen in some individuals, or have been found in some individuals. So here's 4 examples of the mutations that come up in these patients. All right, if single amino acid change can cause this condition, and I've just suggested that broad disruptions of multiple genes are required in order to get the condition, maybe could a single amino acids variant actually broadly disrupt microRNA regulation? And I'm going to show you why we think that could be the case. Because we had, Anna Zinovjeva in the lab, had happened to accidentally come upon some C. elegans Argonaut mutations that also were like that. And one of them was right next door. They were like that in the sense that the mutation had broader effects on microRNAs than would have had if the gene was deleted. How does this work? In wild type, there's 2 Argonauts for microRNAs. In C. elegans, they're redundant. So if you knock out one of them, actually the remaining ALG2 is almost sufficient to take care of the job. However, if these so-called antimorphic mutations, the mutant protein is present, expressed, and runs rogue, let's say. right? So it associates with microRNAs, but it can't repress targets properly, and so it sequesters microRNAs away from ALDS2, and so it kind of, the dysfunctional Argonaut poisons the normal redundancy. So could the Argonaut syndrome point mutations poison the redundancy of other human Argonauts? So we decided we would test that in C. elegans by modeling it. I know I'm running out of time, so if anybody's worried about that, I will accelerate and I'll be done. So we modeled them in C. elegans, and this is, CRISPR-Cas gene editing is so amazing and easy in C. elegans, so we were able to make these mutants and look at them. And two of them had strong developmental phenotypes that we could show were stronger than the null. So this allowed us to suggest perhaps, indeed, the point mutations in humans were causing this condition because they were disabling or poisoning, let's say, the normal redundancy of microRNA Argonauts. Okay, the other ones are milder, and I won't say much about them. And so we then wanted to do molecular phenotyping. What do we want to know? We want to know what's the microRNAs that are expressed and associated with these proteins. And also want to know what the gene activity impact is. And we use ribosome profiling, which allows you to quantitatively measure and identify across the genome all the sequences that are associated with ribosomes. And so for each gene, you can get a measure of how active those mRNAs are on ribosomes. At the same time, you can quantify the total amount of each message. And that gives you, for each message, thousands of which you're measuring all at once, a translational efficiency. So what do we basically find? And this is where I'm going to finish up. We basically found that the different mutations in C. elegans, modeled in C. elegans, can have distinct effects on the microRNA functionality in that animal. Some microRNAs can be affected in one mutant and not in others. Also, the downstream disruptions of gene expression can be quite distinct with these two mutations. And so that also allowed us to take the liberty of considering that maybe the manifestation of these mutations in patients being so different amongst patients between different alleles could in part be due to the differences in the downstream protein balance or imbalance that's being produced. And one of the most amazing things about this is that we found that focusing on targets of microRNAs that are disrupted in both mutants, that those targets are disrupted differently. Primarily in the H751L, those targets are disrupted on the level of translational efficiency and not mRNA abundance, vice versa for the other one. So we think that this offers a basic sciences opportunity to investigate, basically by using genetic approaches and now combining with biochemical and structural analysis, which is going on in other laboratories, we can make some really interesting inferences about mechanism. Now, so there's also, however, the question of what can these studies in C. elegans tell us about the human condition? And I think it's not much in terms of particulars. That is because, remember, humans have many more microRNAs than C. elegans, and I've already showed you how the target repertoire of even the conserved microRNAs can be very different across species. So we don't think it's the particulars of the gene expression disruptions that are downstream that we observe in C. elegans that could be, informative about the human situation, but it's more the character of them and the differences between mutations that we can resolve in the experimental system. And this is my last slide. Argonaut syndrome is a very, human phenomenon. It involves patients, families, clinicians, support teams, and so forth. And it's the patients that are gifting to us the identity of their mutations that allows us to do experiments, genetic or otherwise, and make inferences about fundamental mechanisms that are going to apply to how Argonauts and microRNAs work in all sorts of contexts. But at the same time, of course, we're hopeful and optimistic that we can learn enough from these studies to be able to inform the disease. So in my last 15 seconds, I'm saying thank you to you all for being here. Rosalind Lee and Rhonda Feinbaum for discovering the Lin4. For Gary, for sharing everything we've shared over the years, but most of all, to Candy Lee for everything. Thank you.
Okay guys, that was one of the lectures in medicine. अरू लेक्चर पनि हुँदैछ, इकोनोमिक्स, फिजिक्स, केमिस्ट्री, अर्को अझ एउटा लिटरेचर, त्यसमा पनि लेक्चर हुनेवाला छ। बट आज चाहिँ मेडिसिनमा थियो एन्ड here it is. First half of the lecture, first half.
I now have the great pleasure to introduce this year's Nobel laureate in physiology or medicine, Dr. Gary Ruvkun, our second star. Gary Ruvkun was born in Berkeley, California. In 1973, he received a Bachelor of Arts with a major in biophysics from the University of California, Berkeley. In 1982, he received his PhD from Harvard University after studying bacterial nitrogen fixation genes in the laboratory of Frederick Osseber. Dr. Ruvkun then started his postdoctoral research jointly between the laboratories of Walter Gilbert of Harvard University and Robert Horvitz at the Massachusetts Institute of Technology, remaining there until 1985. He then became an assistant professor at Harvard University and started his own laboratory at Massachusetts General Hospital. Gary Ruvkun is now at Massachusetts General Hospital and professor of genetics at Harvard Medical School in Boston, Massachusetts, USA. Dr. Ruvkun, we are very much looking forward to your Nobel lecture entitled A Vast and Ancient Hidden World of MicroRNAs Across the Eukaryotes. Please join me in welcoming Dr. Ruvkun on to the stage.
Okay guys, another laureate is joining us for continuation of lecture in medicine or physiology, who was awarded Nobel Prize 2024 award for physiology and medicine. So I think it's Gary. Gary. So I'm here, but I'm not in my live end because I have some other works to do.
Am I on? Yes. I thought I might have unplugged. It's really wonderful to get this kind of reception. It's been
Okay. I have to end live today also because I have some other works to do as well. But you can continue watching this lecture on your own. Nobel lecture in medicine or physiology. Okay. मैले लिंक च्याटमा ड्रप गरिरहेको छु. So if you want to continue watching it, please त्यो लिंकमा गएर you can watch. I have to end my live today. Thank you very much for watching. Also subscribe to our channel. अनि I think Nobel Prize को चाहिँ अर्को लेक्चर पनि हामी म लाइभ गर्ने भएको छ. म सोच्छु म के हुन्छ भ्याउँछु कि नै, एन्ड देन हामी फेरि यदि गरिन्छ भने गरौँ न, हेरौँ न के हुन्छ. So thank you very much. Bye bye.